Welcome to EPG Pathshala. I am Anna Sinrang, Assistant Professor in the Zoology Department, Dolatram College, University of Delhi. Today I'll be talking on the module Hypothalamic Hormones. The objective of this module is to help understand the structure of the hypothalamus, definition of the hypothalamic hormones, different types of hypothalamic hormones and their function. Also to understand the role of the hypothalamus in the regulation of endocrine signaling system. To begin with the introduction, all vertebrate animals have many body systems such as circulatory, respiratory, digestive, immune, excretory, endocrine, nervous system, etc., working in coordination to maintain and sustain the individual organism. This coordination is regulated by the nervous and the endocrine system through neurotransmitters and hormones respectively. Further, these two systems are integrated by an eminent part of the brain called the hypothalamus. In Greek, it means under the thalamus. So the hypothalamus lies below the thalamus and above the pituitary gland, forming the floor and ventrolateral wall of the third cerebral ventricle. The hypothalamus stimulates endocrine signaling system, the system comprising of endocrine glands, their secretion, the hormones, their function in response to the information received through neurons from all parts of the body via the production and release of neurohormones called the hypothalamic hormones. These neurohormones cascade to either the release or inhibition of pituitary hormones, a class of hormones pivotal to endocrine signaling in the body. This concept of neurohumoral relation between the hypothalamus and the pituitary gland is majorly attributed to the work of Geoffrey W. Harris. Before moving on to the different types of hypothalamic hormones, let us first understand the structure of the hypothalamus. Coming to the structure of the hypothalamus can be broadly divided into two parts. 1. Subdivisions and nuclei. 2. Neurosecretory cells of the hypothalamus. First, let's have a look at the subdivisions and nuclei. The hypothalamus has feigned regions with distinct nuclei regulating homeostatic functions of the body. Some of the major hypothalamic nuclei are preoptic nuclei, anterior hypothalamic nuclei, paraventricular nuclei, dorsomedial nuclei, posterior nuclei, mammillary nuclei, ventromedial nuclei, arcuate nuclei, supraoptic nuclei, suprachiasmatic nuclei. The hypothalamus, though small in size, about the size of an almond, plays a crucial physiological role mediating the endocrine, autonomic and behavioral functions. Its dorsal border is demarcated by the hypothalamic sulcus, whereas the ventral side is exposed, extending from the optic chiasma to the mammillary bodies. Three regions are differentiated on the ventral surface of the hypothalamus, starting from the anterior limit of the optic chiasma to the posterior limit of the mammillary gland. 1. Supraoptic or anterior region corresponding to the optic level. 2. Tuberal or middle region corresponding to the region of the median eminence or tuber cinereum. 3. Mammillary or posterior region corresponding to the level of the mammillary bodies. Supraoptic region is the most important division, lying above the optic chiasma and holding three important nuclei: supraoptic nucleus, paraventricular nucleus and suprachiasmatic nucleus. Supraoptic nucleus contains nervous cells that produce antidiuretic hormone. Paraventricular nucleus contains nervous cells that produce oxytocin predominantly and corticotropin-releasing hormone. Suprachiasmatic nucleus is located above the optic chiasma and regulates circadian rhythms. Tuberal region is divided into the medial and lateral part, lateral hypothalamic area, by a plane passing through the fornix. The median part holds two nuclei, ventromedial and arcuate nucleus. The ventromedial nucleus controls eating and are referred to as satiety center. Bilateral lesions of the ventromedial nucleus leads to overeating. In contrast, bilateral lesions in the lateral hypothalamic area leads to anorexia and are referred to as feeding center. The arcuate nucleus, also known as infundibular or periventricular nucleus, seemingly holds neurons that regulate the endocrine functions of the anterior pituitary gland or adenohypophysis. Neurons with endocrine functions are also found in the nuclei of preoptic region and other tuberal nuclei. The mammillary region contains posterior hypothalamic and mammillary nuclei. The cells of the posterior hypothalamic nucleus seemingly appear to have an influence on thermoregulation. The mammillary nuclei, though anatomically forms part of the hypothalamus, no correlation with autonomic and endocrine functions have been observed and are rather believed to influence memory. Coming to the neurosecretory cells of the hypothalamus, neurosecretory cells of the hypothalamus are neuroendocrine cells that receive neuronal input, the neurotransmitters from nerve cells from all parts of the central nervous system and transduce it into hormonal secretions which are released into the bloodstream, whereby bringing about neuroendocrine integration. More precisely, they produce prohormones in their cell bodies which are translated into active forms during axonal transport and stored at the axon terminals in vesicular granules until exocytosis occurs by depolarization of the plasma membrane. Structurally, they are like that of the typical neurons with dendrites, axons, and terminals, but unlike that of the typical neuron, they lie adjacent to blood capillaries and release their products into the circulation. Two well-known neurosecretory cells of hypothalamus are magnocellular and parvocellular. Magnocellular neurosecretory cells are large neuroendocrine cells with long axons that terminate into the posterior lobe of the pituitary gland, the neurohypophysis. They are of two types, oxytocin producing and vasopressin producing neurosecretory cells, though some can produce both hormones. Parvoneurosecretory cells are much smaller neuroendocrine cells with shorter axons terminating at median eminence. The release of their products, corticotropin-releasing hormone and other hormones, into the capillary network in the median eminence, from where they are conveyed to the posterior lobe of the pituitary via the hypophyseal portal system. The hypophyseal portal system is a system of blood vessels in the brain that connects the hypothalamus with the anterior pituitary. Its main function is the transport and exchange of hormones to allow a fast communication between both glands. Now moving on to the hormones of the hypothalamus. The major hormones produced from the hypothalamus, that is the hypothalamic hormones, are thyrotropin-releasing hormone or factor, abbreviated as TRH or TRF respectively, corticotropin-releasing hormone or factor, abbreviated as CRH and CRF respectively, dopamine, abbreviated as DA, growth hormone releasing hormone, abbreviated as GHRH, gonadotropin releasing hormone, abbreviated as GnRH, gonadotropin inhibitory hormone abbreviated as GnIH, somatostatin abbreviated as SS, oxytocin abbreviated as OXY or OXT, vasopressin or antidiuretic abbreviated as antidiuretic hormone ADH. These hypothalamic hormones are produced by the cell bodies of the neurosecretory cells, which are unevenly distributed and aggregated to form the many nuclei of the hypothalamus. The hormones, once secreted, are transported through the axon of the neurosecretory cells to either the posterior lobe of the pituitary gland, neurohypophysis, which includes the hormones oxytocin and vasopressin, or to the median eminence, which comprises the rest of the hypothalamic hormones. Oxytocin and vasopressin hormones are stored as neurosecretory granules in the posterior lobe of the pituitary gland and extruded into the circulation when needed. The rest of the hypothalamic hormones are transported from the median eminence to the anterior lobe of the pituitary gland, the adenohypophysis, where they regulate the release or inhibition of pituitary hormones. Let us now have a look at the individual hormones. First, let's have a look at the thyrotropin-releasing hormone or factor. Thyrotropin-releasing hormone is also known as prolactin-releasing hormone. The hormone is produced by parvocellular neurosecretory cells of the paraventricular nucleus. It was first sequenced and synthesized by Roger Guillemin and Andrew V. Shelley in 1969 and were awarded Nobel Prize in Medicine in 1977 for the same. The polypeptide precursor synthesized is a 242 amino acid polypeptide, which undergoes a series of enzymatic reactions leading to the production of the mature tripeptide, thyrotropin-releasing hormone, which probably regulates body temperature. The secreted thyrotropin-releasing hormone through the hypophyseal portal system reaches the anterior pituitary gland, where it stimulates thyrotroph and lectotroph endocrine cells to unleash thyroid-stimulating hormone and prolactin. Thyroid-stimulating hormone targets the thyroid gland to release thyroid hormone required for metabolism, growth, and development. Prolactin targets mammary gland for milk production. Next, corticotropin-releasing hormone or factor. Corticotropin-releasing hormone is a 41 amino acid polypeptide produced during stress by the parvocellular neurosecretory cells of the hypothalamic paraventricular nucleus. It was discovered first by Weil et al. In 1981 in sheep. It is procured from 196 amino acid preprohormone, reaches the anterior lobe of pituitary via the portal system and stimulates the corticotroph endocrine cells to secrete adrenocorticotropic hormone. Adrenocorticotropic hormone stimulates adrenal glands to release corticosteroids, which help regulate metabolism and probably play a role in immune response. Corticotropin-releasing hormone production in the hypothalamus is stimulated when there is a decrease in the secretion of glucocorticoids from the adrenal cortex. Glucocorticoids are a class of corticosteroids, which are a class of steroid hormones with anti-inflammatory and immunosuppressive actions. Next, dopamine or prolactin inhibiting hormone. Dopamine is an amine synthesized from dihydroxyphenethylamine by the removal of carboxyl group. It was George Berger and James Evans who first synthesized the hormone in 1910. It acts as a precursor for norepinephrine or noradrenaline and epinephrine or adrenaline. It is produced by the neurosecretory cells of the hypothalamic arcuate nucleus. It inhibits the secretion of hormone prolactin and hence referred to sometimes as prolactin inhibiting factor or hormone or prolactostatin. Disruption of dopamine secreting cells causes Parkinson's disease. Now moving on to the next one, growth hormone releasing hormone. The growth hormone releasing hormone has two active forms, 44 and 40 amino acids. It is produced by the cells of the hypothalamic arcuate nucleus and released into the anterior lobe of the pituitary gland via the hypophyseal portal system. In the anterior lobe of pituitary, it stimulates somatotroph endocrine cells to produce growth hormone. Growth hormone regulates the metabolism of proteins, lipids, carbohydrates, and indirectly promotes growth. The major target organs of growth hormone are the liver and the adipose tissue. Next, we have the gonadotropin-releasing hormone. Gonadotropin-releasing hormone is synonymous with follicle-stimulating releasing hormone and luteinizing hormone releasing hormone. It is a peptide of 10 amino acids secreted from the preoptic nucleus of the hypothalamus. It stimulates the secretion of luteinizing and follicle-stimulating hormone from gonadotroph endocrine cells of the anterior lobe of the pituitary gland. These hormones stimulate the production of estrogen and progesterone in the females and testosterone in males. Gonadotropin releasing hormone is necessary for normal sexual physiology in both males and females and are released in periodic pulses after every 1 or 2 hours. High level of male and female hormone, that is testosterone, estrogen and progesterone, inhibits the production of gonadotropin releasing hormone by the hypothalamus. Next, gonadotropin inhibitory hormone. The gonadotropin inhibitory hormone is a hypothalamic dodecapeptide discovered in the Japanese quill in 2000. It was named so due to its gonadotropin inhibitory property in the culture quill anterior pituitary gland. It is primarily produced by the neurosecretory cells of the paraventricular nucleus in birds and from the dorsomedial hypothalamic area in mammals. Gonadotropin inhibitory hormone directly inhibits gonadotropin release, unlike that of sex steroids and inhibin that do so via gonadal feedback. Next, somatostatin. Somatostatin is synonymous with growth hormone inhibiting hormone. It has two active forms, one of 14 amino acids and the other of 28 amino acids. It is produced by neurosecretory cells of the hypothalamic ventromedial nucleus and then released to the anterior pituitary gland via the hypophyseal portal system. It inhibits the production of growth hormone from somatotroph endocrine cells in the anterior lobe of pituitary gland. It also inhibits the secretion or release of thyroid stimulating hormone. Somatostatin is also secreted by the pancreas and the intestine, inhibiting the production of insulin, glucagon and varied other hormones. Next, oxytocin. Oxytocin in Greek it means quick birth, is a neuropeptide hormone composed of 9 amino acids produced by mechanocellular neurosecretory cells of the paraventricular and supraoptic nucleus and stored in the anterior pituitary gland. It is released into the bloodstream in response to stretching of the uterus and cervix during parturition and suckling of nipples by the baby during breastfeeding. It brings about the contraction of the uterus and ejection of milk. These properties were discovered by Sir Henry Howlett-Dale in 1906 and Oates and Scott in 1910 respectively. It was elucidated and sequenced by Du Vigneault and was awarded a Nobel Prize in 1955. Next, vasopressin. Vasopressin is also known as antidiuretic hormone composed of 9 amino acids. It regulates reabsorption of water in the kidney and constriction of blood vessels, thereby increasing blood pressure. It is found in most mammals and contains arginine in most species and hence referred to as arginine vasopressin or argipressin. It is produced by the magnocellular neurosecretory cells in the hypothalamic, paraventricular and supraoptic nuclei. It travels down the axon and is stored in the posterior pituitary gland bound to a protein. It is released into the bloodstream when the blood osmolarity increases. Coming to the regulation of the hypothalamic hormone production. Secretion of the hypothalamic hormones are governed by various internal and external stimuli such as age and sex of the individual, sleep-wake cycle, photoperiod, stress, feeding, etc. A significant regulatory mechanism of hypothalamic hormone secretion or suppression is the negative or positive feedback from the end products or hormones released from the target endocrine glands of specific trophic hormones of the pituitary gland or the tropic hormones of the pituitary themselves. Majority of the hypothalamic hormones are governed by negative feedback. Oxytocin production is governed by positive feedback. Now to summarize the module, hypothalamic hormones are neuroendocrine hormones produced by the neurosecretory neurons of the hypothalamus. The hypothalamus acts as the integration center for the nervous and the endocrine system. There are about 8 hormones produced by the hypothalamus. Thyrotropin-releasing hormone stimulates thyrotroph cells in the anterior lobe of pituitary gland to secrete thyroid-stimulating hormone. Corticotropin-releasing hormone stimulates corticotroph cells in the anterior lobe of pituitary gland to secrete adrenocorticotropic hormone and melanocyte-stimulating hormone. Dopamine, it inhibits the secretion of hormone prolactin. Growth hormone-releasing hormone stimulates somatotroph cells in the anterior lobe of the pituitary gland to secrete growth hormone. Gonadotropin-releasing hormone stimulates gonadotroph cells in the anterior lobe of pituitary gland to secrete follicle-stimulating hormone and luteinizing hormone. Gonadotropin inhibitory hormone inhibits the activity of gonadotropin-releasing hormone neuron of the hypothalamus and the gonadotrophs of the anterior pituitary gland. Somatostatin, it is also known as growth hormone inhibiting hormone. It inhibits the production of growth hormone and thyroid stimulating hormone in the anterior lobe of the pituitary gland. Oxytocin, it stimulates the contraction of the uterus and ejection of milk from the breast. Vasopressin or antidiuretic hormone, it helps in reabsorption of water in the kidney tubules and maintains blood volume. Majority of the hormone, that is thyrotropin-releasing hormone, corticotropin-releasing hormone, dopamine, growth hormone-releasing hormone, gonadotropin-releasing hormone, gonadotropin inhibitory hormone, somatostatin are released into the anterior lobe of the pituitary gland via the hypophyseal portal system, where they stimulate or inhibit the endocrine cells to release respective hormones according to the internal and external environmental needs of the body. Oxytocin and antidiuretic hormone are stored as vesicular granules in the posterior lobe of the pituitary gland and released into the general circulation according to the need of the body. So this is all about the hypothalamic hormones. I hope it will be of some help. Thank you.